so now that we know the overall shape and structure of DNA, what they're also determined, Watson and Crick were able to determine is how DNA makes copies of itself. So let's take a look at DNA replication. In DNA replication, this is a synthesis of a new DNA molecule. And this type of replication is what is known as a semi-conservative replication. That's where one of the old strands is present in each new daughter molecule. And so here, the old strand is used as a template. So with the template, you make a complementary shape to itself. So right here in the middle is trying to show the semi-conservative replication. And all we need to look at is right here. So here's the old DNA molecule, 2 strands in the dark blue. And so what we're going to do is we're going to separate those strands from each other and use that old strand to make the new strand, all right, which is shown there in light blue. Okay, so let's look at the steps in here. The first step is called unwinding. And here an enzyme called helicase breaks the hydrogen bonds that pair the nitrogenous bases. So if we go back to this picture here, helicase acts as a zipper and it just unzips these 2 strands from each other and they're going to separate from each other. All right, so that's what we see here. We separated those 2 strands from each other. All right, next is complementary base pairing. So new complementary base pairs are put into position, and this is done by an enzyme called DNA polymerase. So as we can see here, C is put in with G, T is put in with A is put in with T, G with C, and so on. Okay? Next is joining, and here a different enzyme called DNA ligase joins the new nucleotides together to form a new strand. So when we first put these guys into position, that C is not bound to that T, so that's what ligase does. It binds those together, making that new strand. All right? So now at the end, we have 2 molecules of DNA. Each of these molecules is made of 1 old strand and 1 new strand. So let's take a look at the rate of replication. So if we look at eukaryotic cells are cells that have a nucleus. So here, about 50 nucleotides are put into position per second. Now, there are several origins. Now, origins are beginning points, all right? So that's what they're trying to show. And these things are called replication bubbles, all right? So it'll move bidirectionally as well in both directions until, we have 2 DNA daughter molecules. And it takes a few hours for eukaryotic cells for this. Now, in this process, about 1 in 100,000 nucleotides are put into the wrong place. So we have a mismatched nucleotide there, all right? So that is, if that would stay, that would be a mutation. Now, there is a proofreading step that eukaryotic cells have. So they go back through, make sure, this is done by DNA polymerase, to make sure that, A is with T and G is with C and so on, all right? So even after that proofreading step, there is about 1 in a billion mismatched nucleotides or mutations that have occurred, all right? Now, when we look at the entire human DNA, our DNA is made of about 3 billion nucleotide base pairs. So what that means is every time we make a copy of our DNA, there's roughly about 3 mistakes in there. Now, later on, we're going to find out a lot of this is going to occur in places where it doesn't matter. But we'll get to there. All right, now let's take a look at prokaryotic cells. With prokaryotic cells, all right, about 50 nucleotides are put into position, about 500 nucleotides are put into position per second. They only have 1 origin, but because they have much smaller DNA, it only takes about an hour, all right? Now, they do have a slightly higher mutation rate. They do have a proofreading step, but a slightly higher mutation rate, okay? So that's what we see with prokaryotic cells. Now, viruses use the machinery of that cell that they have infected, all right? And so viruses have much shorter DNA, but they lack a proofreading step. And because of that, viruses can mutate fairly readily, and all they need is 1 cell. And this is why it's so hard for us to get a handle on the cold viruses and flu viruses, Because all they take is to get into one person, they can mutate in that one person, and you have a new strain that you have to develop an immunity all over again to.